So this is a video on net charge calculation of amino acids. So let's say in a problem you are given a tripeptide sequence or peptide sequence of 3 amino acids or 4 amino acids or 5 amino acids, and you want to calculate the net charge on the peptide sequence or the amino acid sequence at a certain pH. So let's say in this case we want to calculate the net charge on these 3 amino acids at pH of 5. So we know that around physiological pH 7.4. So if you know that um, from the Henderson-Hasselbach equation, we can calculate we can find the relationship between pH and pKa. So if pH is greater than pKa, that means that we have deprotonated form of an amino acid present. So we will have the concentration of deprotonated form higher than the protonated form. So these amino acids will exist um, in their deprotonated form. And if pH is less than the pKa groups of the N terminus or C terminus or these R groups, then we will say that the protonated form is the predominant one. So they will exist as the protonated form. Okay. So this is the idea behind the Henderson-Hasselbach equation, which lets us calculate pH or pKa values if you are given pH, or we can also use these. This equation is really important in calculations of titrations and buffers and indicators. But here we are going to talk about amino acid net charge calculation. So once we know this relationship that if pH is higher than pKa, we will have the deprotonated form. If pK is higher, we will have the protonated form. Everything becomes really easy. So let me show you how. So let's say let's consider one amino acid first. So let's say we want to consider methionine. And in one amino acid, we don't have peptide bonds, right? We don't have the amide COONH bonds. We only have one amino acid. So this is how amino acids usually exist. So this is the R group of, let's say, methionine, right? And this is how amino acids usually exist in their native state. So around physiological pH. So we know that the C terminus, the carboxy group, has a pKa of around 2. And this is for most, all of the amino acids. Some have below 2, some have like 1.7. Let's say for cysteine, the pK value for the carboxy group is around 1.7, but for some of them it's more than 2. So let's say for histidine, it's more than 2, but we can approximate it to around 2. Okay. And for the N terminus, for all amino acids, it's around 10. So the pK value of the N terminus would be around 10. Okay. So this is the basic idea that we use in the calculations of the net charge for single amino acids or dipeptides, tripeptides, or we could have any number of amino acids for net charge calculation. So this is the basic idea. So around pH 5, what will happen is that this will exist in the deprotonated form. Why? Because pH 5 is greater than the pKa of the carboxy group. So 5 is greater than 2. So it will be found in the deprotonated form, right? But for N terminus, its pKa 10 is greater than pH 5, so it will be found in the protonated form. So that's why it's protonated, it's got a plus charge. So this is usually the normal or native configuration of amino acids when we don't have a highly acidic or highly basic environment. Let's say if we have pH 12, then this would still be deprotonated, but this will also become deprotonated because now pH 12 is greater than its pKa value of 10. So it will lose its positive charge and it will become deprotonated. So in that case, amino acids will exist in the net negative charge form. Okay, so that's the basic idea. If we have, on the other hand, a highly acidic environment, if we have pH 1, then what will happen is that this will go back to its protonated form, and this will also become protonated because now both of their pKa's is greater than the pH 1 environment in the highly acidic environment, so they both become protonated. So this will also become protonated, and in that case amino acids exist in the positive positively charged configuration. Okay, so that's the basic idea you need to understand for N terminus and also for the C terminus for amino group and carboxy group. Once we are once we can understand this, anything related to net charge calculations for amino acids becomes really easy. Okay, so this is the basic idea. Now let's look at the problem that we are given. Now in this case we have methionine, isoleucine and aspartic acid. So as I told you, between that range 2 and 9, so I told you that carboxy terminus, this exists, this has a pKa of around 2 and the N terminus has a pKa of around 10. So any pH between these two pKa values, the amino acids will be found in their native configuration, in the charge that they are usually in. So unpolar uncharged amino acids like methionine, isoleucine will be in their neutral form. But charged amino acids, aspartic acid and glutamic acid, these are the acidic ones, so they'll be found in the negative configuration. But histidine, arginine, and lysine, they are the basic amino acids, right? So these three will be found in the positive plus charge configuration, right? So that's the basic idea. Now let's look at, now we know that pH 5 is between 2 and 10, right? So we know that methionine and isoleucine will be in their neutral form and aspartic acid will be in the negative form. So let's go ahead and calculate the uh, overall net charge on this tripeptide. So if I draw the backbone structure of this tripeptide, it will look something like this. So in this case, now we are not considering one amino acid. Now we are considering a tripeptide. So we'll have peptide bonds, right? So if I go ahead and draw that. Actually, let me erase this and then. So we know that the pK of the C terminus is 2 and for N terminus it's 10. Let me write it here. So C terminus has a pK of around 2 and N terminus or the amino group has a pK of around 10. Okay. Now let's draw the backbone structure of this tripeptide. Let's see how we can work out the negative charge. So I start with the N terminus. We always start from the N terminus and go towards the C terminus. So we'll start with methionine and this is its N terminus and let's denote its R group like that. Okay. So I'll start from here. So this is, let's say, the R group of methionine and we know it's neutral at pH 5. We checked it from the pK values. Okay. And this is your peptide bonds connecting the amino acids. And this is, now we are in the second amino acid. So now it will be the R group of isoleucine. Now this is another peptide bond. And now I'll have the R group of aspartic acid. So only in this case aspartic acid, as we checked, is charged. Its R group is charged. The rest of the methionine and isoleucine R groups are neutral. So this will be negative, so I'll put a negative sign over here. And aspartic acid is D, denoted by D. So I'll put a negative charge over there. And we have this. So we already know that in this case, even though we have three amino acids, we have a tripeptide, right? We have methionine, isoleucine, and aspartic acid. But we don't have three N terminus values and three C terminus values. Why? Because we have peptide bonds in this case. So you have to account for peptide bonds. So if you're confused, you should draw out the peptide backbone structure of the peptide given to you. So the knowledge of peptide bonds and the backbone structure of peptides, it comes really handy in this case. It's really helpful. Because if you go ahead and just calculate the net charge on these three amino acid tripeptide, and you, by mistake, let's say you consider that all of them have their own N terminus and C terminus, so you don't account for these peptide bonds, then you'll get the wrong answer. Because we only have in this case the N terminus over here the terminal N terminus, and over here we have on the right side we have the C terminus that comes from aspartic acid. So we only have two to consider over here because rest of the other two are peptide bonds. So that's the trick that you need to understand. Okay, so now we saw that because C terminus at pH 5 will be negatively charged, because why? Because because at pH 5 its pK is 2, it's less than the pH, so it will exist in the negative form, right? But for N terminus its pK is greater than pH, so it will be in the protonated form. So that's why it's protonated. So this is plus charge, this is negative. So we have a balance of charges on the two terminal groups, amino group and carboxy group, and we have a negative charge over here, right? For our group, for aspartic acid, right? So then overall we have a net -1 charge. So that's your answer, -1. So that's it, we are done. But if you need to consider that, if you need to consider the aspartic acid and its R groups, because for charged amino acids, their R groups have their own pKa values. So for methionine and isoleucine, we don't consider the pKa values for their R groups because they are neutral. But for charged amino acids, we consider their R group pKa values. So if you want to take a look at aspartic acid, it's 3.86, right? That's its pKa value for its R group. So this R group has a pKa value of 3.86, okay, around 3.86. So we can take that into account and do calculations on this R group as we did on the carboxy and N terminus. So we can see that its pKa is less than the pH. So it will exist in the deprotonated form. And that's why it has a negatively charged. If the pH would have been below this pKa value, let's say around 3 or 2, which is below 3.86, then it will be protonated and it will lose its negative charge. Right? That's the trick. So we took into consideration everything about this peptide and we came to the conclusion that the net charge is -1. So that's how you calculate the net charge of a tripeptide given to you at a certain pH. So it's all about, there are 3 tricks. You have to recognize the peptide bonds. So I'll say in 3 steps, if you want to tackle a problem like this, what you'll do is that first you will draw the peptide structure. So draw the structure. This will give you the idea that where the R groups are located. And there are peptide bonds that you only need to consider in any case. Only one carboxy terminal and one amino group terminal. So even if it was 5 amino acid peptide or 6 amino acid peptide, in that case also you'll have only one charge C terminus to consider and one amino acid N terminus to consider. Okay, so draw the structure. And the second thing that you only need to account for are the pKa values. So that's the only 2 things that you need to consider. For pKa values, as I told you that for charged amino acids like aspartic acid or glutamic acid or the basic amino acids, lysine, histidine, arginine, for them you need to consider their R group pK values as well. So for them, for these charged amino acids, these 5, acidic and basic, and also cysteine and tyrosine, they are not charged, but their R groups are ionizable. So for these 7 amino acids, you'll have 3 pKa values to consider. Okay, but if they are inside the peptide sequence and they are surrounded by peptide bonds, then you don't need to consider. Then in that case, in any peptide, I told you that you'll only consider the C terminus, one C terminus and one N terminus. So the problem in that case becomes very easy, and you only need to take care after that for the R groups present. So that's it. We'll also look at the calculations for more complex amino acid peptide sequences. In another video, I'll show you how to calculate the net charge on, let's say, 5 or 6 amino acid peptide sequence. Okay, so I hope it was helpful. Thank you.